Hello, Professor. I'm gonna be doing the weekly review for week 9, weekly review 9, and I'm gonna take my notes from the introduction, notes of the class, and also from Brightspace. And I'm gonna be answering the questions now. So I was just checking to see if you could read that. I didn't want to record the video all in the opposite point of view. That would be so useful. Well, okay. First, we're gonna start talking about the figure 2D, the one with the CHX. And the first question is, what type of experiments we do in the figure 2E, and we do Western blot, and we also do the qPCR. So for figure 2E, we're gonna do Western blot and qPCR, which we do Western blot to quantify proteins, and we do qCPR to quantify RNA. So, first, we're gonna describe how is the Western blot going to work. We're gonna do a little cartoon of this whole thing. So first, we're gonna have the sterile fed HEP, because the cells that we're gonna use HEPG2 cells, which are sterile fed, and they are pre-incubated in this CHX. I think it's 10 milligrams. Yeah, whatever. Sorry. Doesn't really matter how much, but it's pre-incubated in CHX for 1 hour. So we're gonna have this plates. We're gonna have 4 plates. Okay. Yeah, we're gonna have 4 plates, and after we have the 4 plates, we're gonna leave them for 24 hours. 2 of these plates is gonna have vehicle, and 2 of the plates are gonna not have any vehicle. But however, all of them are gonna have MK2206. Well, not all of them, because some of them do not reach MK2206. Some of them, 2 of them are gonna be treated with MK2206, and 2 of them are not. As it's explained in the graph, yeah, pretty sure. So we wait 24 hours, and this, because we're gonna harvest the cells, and the cells are gonna be split into 2. These cells are gonna split into 2, and that's why at first I thought it was 8 plates, but it's actually 4 that splits into 2. That's why in the graph you can see like 4 bars, but actually the bars split into 2, each one having CHX, and the other one has not CHX but the vehicle. A little prettier. So we split them, and for the first half of the plates, we're gonna isolate our protein. So we isolate the protein by collecting the cell pellets. So that's like the Eppendorf 2. We're gonna isolate the protein by collecting a cell pellet. When we collect the cell pellet, we unthaw them for like experimentations, and then we're gonna lyse this with buffer, and then we're gonna add more buffer. More buffer is added, and after the more buffer is added, we're gonna do a protein assay. So we're gonna quantify this protein. And after we quantify the protein, we're gonna, wait a second. Or we're gonna run the gel. After we quantify the protein, we're gonna run the gel. So I'm gonna erase this tool. So we have the gel, we run the gel after the protein assay, and after we run the gel, we put the block membrane. So we do the block, and after the block, we can add, so after the block membrane, we add the antibody, that's right. So we add the antibody, primary antibody is gonna be from rabbit, anti-LDLR, and anti-GAPDH, which is the primers that are gonna be in this experiment. And we're gonna, after that, we're gonna wash with some PBS, and we're gonna add we're gonna add another antibody, which is a secondary antibody, which is come from the goat, and it's anti-rabbit. And after we add that second antibody from the goat, we're gonna wash it again with PBS, and then we're gonna go to the blot, the Western blot. So at the end, after doing all of those, we can do the blot completely. For the second half, for the other half of the cells that were split, these go through qPCR. So for qPCR, we're gonna get this. We're gonna get the RNA. So RNA is isolated at first, and then the cDNAs, or complementary DNA, is made out of this RNA, and then we're gonna have this tube here, open the tube with RNA in there, right? I think you can see that. And we're gonna add to this tube, we're gonna add LDLR primer, we're gonna add GAPDH primer, we're gonna add, what else are we adding? So we added the LDLR primer, we add the GAPDH primer. Oh, we add the cybergreen so it can pop out in the gel. And after adding this three, we also add the cDNA. So all this go in here. And after we add all those things to the tube, we can do the qPCR. We can run the qPCR. Because for PCR we need DNA template. qPCR. So, okay. And that's what's gonna happen for those cells. So, and after that, so basically what the MK2206 that we use in this experiment, it's gonna cause to increase the LDLR protein. So the MK22 is gonna increase the LDLR protein, like you can see in the graph, but it's also gonna increase the RNA levels. So, in conclusion, in comparing to the introduction, the intro says that the MK2206 can decrease or lower the cholesterol levels. So, it can decrease the LDLR up in the blood, and that way it can also decrease the cholesterol in the blood. However, in this experiment, the LDLR protein is increased by the MK2206 and also the RNA levels, so I'm guessing the cholesterol would be high in this person. Yeah, that would be it.